When we're studying microbiology, there's certain things we need to keep in mind, okay? Everything, this desk, my hand, my body, this camera, this light, the dust that I'm breathing in, everything is covered in microbes. Not if it's not living bacterial cells, then it's bacterial spores or fungal spores, whatever. So we have to try to maintain some level of sterility. Everything we work with in a lab has to be sterile, because if it's not sterile and things grow, well, is it the stuff we're trying to study that's growing, causing the change in the media? Or is it some outside contaminant that was just there before we got started? Or something that was introduced because we were doing work on it? This is the idea of aseptic technique. Aseptic technique means that while I am doing work, I am going to maintain sterility, meaning I am not going to introduce any unknown outside organism. If I am transferring organism A from a tube to another tube, well, that new tube will only have organism A that I have transferred over. It's not going to get any dust. It's not going to get any bacteria off of my skin or any dust out of my breath from my sinuses or my mouth. It's not going to get anything but organism A. That is aseptic technique. And for a lot of students, that is initially something that's very hard to try to maintain. Because, oh, I only touched it for a second. Well, it only takes a second to transfer bacteria from your fingers to the petri dish, from your fingers to the loop you just sterilized, and now you're getting ready to get bacteria from your plate or your broth culture. Because we're trying to do all our work with what is referred to as pure cultures. A pure culture means that we know exactly what microbial species is present there, and it is only that microbial species. There are no unknowns there. There are no, well, it's this one I wanted to study and maybe 1 or 2 or 10 other things. No. Because if we try to do something that's, work in a lab, characterization, diagnostic microbiology is what we're going to be doing the first, couple of months of this semester in lab. If you don't have a pure culture, your results are not going to make sense. What should have been a negative test because you think it was this suddenly becomes a positive because you introduced some unknown things, one unknown or multiple unknown species. It won't work. We could start off with what are known as enrichment cultures. Okay, if I was to do if in a clinical setting I was to do a swab, oh hey, you've got a wound that's infected. Let me take a swab. Well, that wound is not going to be, infected with just one microbe. There's going to be multiple microbes there. So, what is the clinical lab at the hospital going to do? They're going to do this enrichment technique. Okay, what's the predominant bacteria there? We're going to come up with ways to transfer and try to get just that one so we can run tests on it to identify it and then figure out, what antibiotic resistance, what antibiotic susceptibilities are there. So yeah, in a clinical setting, because I know a lot of you are thinking clinical of some sort, nothing they do in the hospital is pure culture. I have a cut, I have a wound that's infected. It's going to be different species. They're going to try to, they're going to try to go from a multiple species thing through, steps of enrichment to try to get to this pure culture. And if we think back to originally, it was only a few hundred years ago when what we think of as microbiology started. This is post-Hook, post-Van Leeuwenhoek, who, hadn't quite really started microbiology yet because the idea of germ theory and, spoilage or diseases being caused by germs hadn't really kicked in yet. That usually, that's not until about the mid-1800s. But they're still operating under this idea of spontaneous generation. And there were quite a few people who tried to come up with ways to dispel spontaneous generation. Reddy is one. You took the meat, put it in, and flasks, left one flask with meat open, covered another one with cheesecloth. The idea was at the time was the meat, that chunk of meat, would spontaneously become maggots, which would then develop into flies. Well, ready, hey, look, I'm going to cover these, the one that's open, the one that's covered, cheesecloth, so the air still gets into both, but nothing else gets in the one covered in cheesecloth. Well, of course, we now know, flies get on the meat, flies eat, flies lay eggs, eggs hatch, you now have larvae, maggots. But if you have the cheesecloth covering the flask that they can't get down to the meat, well, they're not going to eat, they're not going to lay, there's not going to be any maggots. So there was many people after that who didn't believe ready and still spontaneous generation. You can get life from dead inanimate things until work by people like Pasteur. Pasteur is one of these people, crazy, stupid, smart. Okay, let's just get that out there. He was able to go through and show that certain microorganisms, just because it's a sugar doesn't mean it's a sugar that all of them are going to use. Yes, we see sugars, and at the time, they had differences being able to differentiate certain sugars. But he was able to show, Pasteur, that, hey, yes, they knew optical isomers. They were able to do that much in chemistry. And he's able to show that certain organisms have preferential for certain isomers over others. We now know that, that's going to be genetically driven. They have the genes to be able to use lactate, but not, lactose, but not, galactose, or glucose, but not fructose, and things like that. He was able to show that fermentation, alcoholic fermentation, was a biological process. Yeast, bacteria, took in the sugars, and through, anaerobic means, were able to produce alcohol. Up until then, many people just thought this was a chemical process. You mix all these things together, and then just like in, chemistry class, you mix stuff together and poof, you get something new. No, the making of alcohol only occurred when certain biological cells were present. That work was based on, the work he was doing because he was hired to try to figure out why certain foods were spoiling. Specifically, some vineyards and, I believe it was vinegar makers were trying to figure out why things were spoiling. What's causing these things to spoil? Because, you got to remember, vineyards, these wines and stuff, that's big business. They were losing, cask after cask of, what should have been wine was bad vinegar, and some other people were making vinegar using chemical process, couldn't figure out why what shouldn't be vinegar yet was turning into vinegar, and it wasn't a good vinegar, and it was just all sorts of stuff. So that's where he got into the studying of what we would now call microbiology, and he wouldn't have considered himself a microbiologist. He was a chemist who liked to play with microscopes. One of the things he did and he looked at was this idea of spontaneous generation. You've heard about his gooseneck, experiment since you were a kid. He filled up a couple of flasks with a broth that he had boiled. Or, I'm sorry, they hadn't boiled yet. He put them in the flasks. He used a heat source because if you heat up glass, remember, you can bend it, you can stretch it. So he filled up some flasks with something that wasn't sterile. We hadn't done anything yet to it. Bent this into this gooseneck. The whole idea of this gooseneck is that gases could be exchanged, oxygen in, good air in, bad air out. But because of this bend would capture any dust. The dust particles are not going to be able to go up and around because, there's not a really good breeze that's going to carry them up. This is just a passive, air diffusion here. He boiled this broth to kill anything and everything that could be there, and then he let it cool. There wasn't a passive air exchange. Dust settled down, got captured here in the bend. But because the dust didn't make it up, whatever was on the dust didn't get into the liquid, it remained nice and clear. Some of the flasks he bent forward so that the liquid could go up to where the dust is, and then he would set it back up. So some of that dust would be carried back. Well, within hours, that broth became cloudy. Hey, guess what? Something's on the dust. Something microscopic is on the dust. Now we have microscopic living things causing this broth to spoil. This is what we would think of as nowadays as the definitive nail in the coffin for this idea of spontaneous generation. And the idea to get life, you have to have life. And that life was carried on as microscopic things, organisms, on these particles of dust. He went on, he did a lot of amazing things beyond just, here's how fermentation works, biologically, cells. He didn't go through and do all the work on fermentation. There's been many people after him, but he was the first. It's a cellular process, not a chemical process. He also came up with, he got into vaccines. One of the big ones, rabies. He's the first to come up with the rabies vaccine. Here, this picture, I can't remember the name of this boy. Boy got bit. And this is him showing him, injecting the young boy before he started to show symptoms of. Because that's the thing with rabies. You can't wait until you show symptoms of, because by then it's too late. You've already got the brain damage. But if I get bit today, I have a window of, I think it is like 6 to 8 weeks. I can't remember, which is the incubation period, where if I get the vaccine shots, my immune system kicks in before the disease kicks in, and I stand a pretty good chance of staying alive.